Good day, everyone. My name is Günel Əyyubova, and I'm going to present you the information about the third lecture. It is the nucleus, the structure of the chromatin, cell cycle, types of cell division, cell aging and death, the clinical cytology. The objective of our lecture is to get the knowledge on the structure and microscopic appearance of the nucleus and its components, the nuclear membrane, nucleolus, nucleoplasm related with their microscopic appearance, and about the cell cycle. So these are learning goals. So define differences in the nuclear shape, number according to the cell shape and functional phases of the cell, to define microscopic appearances of the nuclear membrane, to define microscopic appearances of the nucleolus, and so heterochromatin and other components of the nucleus. As we know, the nucleus, it is the largest organelle in the cell, and it is cell's control center. The nucleus contains three major components. One of them, it is the chromatin. It is the storage of genetic information of the cell. Also, the nucleus, it is the site of DNA replication and RNA synthesis and processing. And now we are looking at the nuclear envelope here. That is actually it composed of the inner and outer nuclear membranes with an intervening perinuclear cistern here, perinuclear cistern, which is located between these two membranes, and where the inner and outer nuclear membranes, the regions where the outer and inner membranes fuse, there is the nuclear pores. We have to know that the outer membrane, pay attention here, the nuclear pores here, the outer membrane, it continues into the rough endoplasmic reticulum, and that's studded with the ribosomes. And in the outer membrane, we also have the ribosomes. It means that outer membrane participates also in the protein synthesis, but here produce proteins of the nucleus. Nucleus, it is maintained in its place by the surrounding network of filaments, which maintain the location of the nucleus inside of the cell, the stable location. The filaments that surround nucleus, these are vimentin intermediate filaments that form the support for the nucleus. And here we can see the nuclear pores, which are important for controlling the traffic. Nuclear import and nuclear export signals are recognized. Here nuclear signals are recognized, and selectively transport is permitted. So from the cytosol into the nucleus is transported proteins and polymerases, enzymes, DNA and RNA polymerases, regulatory proteins. From the nucleus to the cytoplasm transported tRNA, messenger RNA, and also ribosomal subunits. Actually, we have to know that here we can see the membranes, outer membrane, inner membrane, and inside of the nucleus we have chromatin, the nucleolus we have here, and all of the content of the nucleus is filled with the nucleoplasm. Pay attention to this illustration. Here we are looking at the chromatin content, and here so the main components of the nucleus, these are the main components of the nucleus are the chromatin and the nuclear envelope that surround nucleus, then the nuclear matrix or nucleoplasm, and nucleolus. These are main components of each nucleus. That is the electron microscope representation of the nucleus, and here we are looking at all of those components just I mentioned. And under the light microscope we have disappearance of the nucleus, and nucleolus we can distinguish inside of it. And so nucleus may have different shapes depending on the cell type. They may have rounded shape and elongated oval shape, like in this case that are muscle cells. In muscle cells, so nucleus may have elongated shape. And also the location of the nucleus may be different, maybe in the center or at the periphery of the cell. And size and microscopic features of nuclei tend to be uniform in tissues. The number of nucleus, usually each cell, it has just one nucleus. But sometimes infrequently dividing cells found in highly regenerating organs. For example, hepatocytes may have two nuclei inside of the one cell. And rarely multiple, for example, osteoclast cells here. Osteoclast cells include a lot of nuclei. These are bone-resorbing cells in the bone tissue. Also, we have skeletal muscle cells, which include a lot of, these are skeletal muscle fibers, which include a lot of nuclei. And also sometimes nucleus may have lobulated appearance, for example, in neutrophils and eosinophils. These are blood cells in which nucleus may have lobulated appearance. The size of the nucleus may be different, and it's changed according to the size of the cells. For example, the oocytes, the female sexual cells, they in the cell, the nucleus is largest. Oocytes are largest cells in the body, approximately 200 micrometer in diameter. That's why the nucleus is also large. Purkinje cells, these are neurons, nerve cells in the cerebellar cortex, and they have also large nucleus. And finally, neurons, neurons in the central nervous system, which are neurons in the brain cortex, for example. These are largest cells. Also, we have granule cells also inside of the central nervous system. They may have small nucleus. Here we can see that nuclear envelope consists of these membranes, two membranes between the perinuclear cistern, we can see. And here we also can distinguish ribosomes on the surface of the outer membrane. But inner membrane, it lacks the ribosomes, and its innermost surface is in contact with the clumps of the heterochromatin here. Then during the nuclear envelope, during the division of cells, it disappears, and at the end of the prophase until the end of the telophase. Here we can see the dividing cell in which we cannot distinguish the nuclear envelope. And then after division, the nuclear envelope is reformed by the rough endoplasmic reticulum during the end of the telophase. The membranes, here we can see again the membranes, out and inner, and under the inner nuclear membrane, we have the network of filaments, of intermediate filaments, which called lamine proteins. The lamines, they are the intermediate filaments which form network here, and there are also in the inner nuclear membrane there is the lamine receptors located here. In the inner membrane we have lamine receptors and several lamine associated proteins receptors that bind to chromosomes and nuclear lamina. So nuclear lamina here by schematic representation, and that is by scanning electron microscopy, we can see that is electron microscope of the outer and inner membranes that's separated by the perinuclear cistern, and these are the nuclear pores are looking here. The nuclear lamina, it lies between the inner nuclear membrane and the peripheral heterochromatin. That's why it helps to stabilize the nuclear envelope, and major components of lamina, they are nuclear lamins, then specialized type of nuclear intermediate filament, and lamina-associated proteins. So there are not just lamins, but also lamina-associated proteins. This lamina composed of three main types of lamins. These are A, B, and C. Nuclear pore, here again we are looking at it. These are pores on the surface of the nuclear envelope, and they are located at sites and in and outer membranes of nuclear envelope fused. So here in these regions, the outer and inner membranes are fused, and they are not opened, and the diaphragm-like structure appears to cross the pore opening. A small dense body is observed at the center of the opening in transmission electron microscope. So it's not open pore. So nuclear pore we can see by specialized techniques such as voltage transmission electron microscope. Additional structural details we can see, we can observe. Actually, that nuclear pore is surrounded by a lot of proteins which together form nuclear pore complex, and here we have to mention that these are here we can see the subunits, 8 subunits, which together they form the network, they form the scaffold of the nuclear pore complex. Here 8 multi-domain protein subunits arranged in an octagonal central framework at the periphery of each pore, and they form a cylinder like structure known as nuclear pore complex. Nuclear pore complex is composed of approximately 50 different nuclear pore complex proteins referred as nuclear porins. These nuclear porins, the central framework is inserted between two cytoplasmic and the nucleoplasmic rings. So we have cytoplasmic, we have nuclear rings. From the cytoplasmic ring, 8 short protein fibrils protrude into the cytoplasm, and nucleoplasmic ring complex anchors a basket that assembled from 8 thin long filaments joined distally by 30 to 50 nanometer diameter terminal ring. So they form the nuclear basket. The cylinder-shaped central framework encircles the central pore of nuclear pore complex. Here we also can see it. And so the meshwork of intermediate filaments associated with nuclear site of envelopes consist of the nuclear lamins. These are nuclear lamina, which composed of these lamins, that is located under the inner nuclear membrane, and that is a nuclear pore complex. As I told you, that out in this pore region, outer membrane continues into the inner membrane, so they are not interrupted in both sides. They just continue into each other. And in the center of the nuclear pore complex, we have central channel. And then here we are looking at the nuclear pores. And so the localization and relationship of the nuclear lamina with chromosomes, here we can see, and in this region. So now we will look at the nuclear pore complex by the illustration which you have in your atlases. Nuclear pore complex, it's composed of a nuclear pore and associated glycoproteins. Here we have 8 spoke-like structures which called spokes. These are they are purple colored structures which are convex spokes. There are 8 spokes which form the wall of the cylinder like structure. And these 8 spokes are joined to each other via rings, which have green color here. We have cytoplasmic ring, luminal ring, which faces the perinuclear cistern, that's why it's called luminal. Then we have nucleoplasmic ring. So 3 rings we have here, which join these spokes to each other. Besides, we have inner spoke ring that is located around the central canal, that is central part, which is like small cylinder that is located inside of the large cylinder. And we have to know that the transportation of substances is taking place through the central this part. Just if the diameter of the nuclear pore complex is approximately 130 nanometer, then the diameter of the central channel is 10 nanometers. And here we have around the central canal we have inner spoke ring. Then from the nucleoplasmic ring arise these radial filaments that radiate toward the nucleoplasm, and they join each other as a terminal ring. Just I mentioned in previous illustration also we saw that is the nuclear basket is formed. Pay attention, that is the basket here and also here. So the nuclear pore complex is made of 2 protein rings and octagonal in an octagonal organization. From the cytoplasmic ring, long filaments penetrate the cytosol. These are cytoplasmic filaments. And from the inner nuclear ring arise these nucleoplasmic filaments that constitute a basket-like structure. And so that is the structure of the nuclear pore complex. Here we can see by scanning electron microscope these pores in the nuclear membrane, in the nuclear envelope. So why we have these nuclear pores? Actually, they are needed for transportation of substances, and the transport here is something complex process. We have to mention, we have to know that there are many nuclear pores. These are many small octagonal apertures that perforate the nuclear envelope, and they mediate active transport of proteins, nucleoproteins, and RNA between the nucleus and the cytoplasm. And here we have to take in mind that all of the components, all of the substances which are needed, which are going to enter the nucleoplasm, they must have the region which is nuclear localization signal region. Then they should bind to the transporters which called importins. So they will bind to importins. Afterward, this structure will be able to enter the nuclear pore. And because in the nuclear pore complex that importins will be recognized, and then afterward this complex will enter the nucleoplasm. Inside of the nucleoplasm we have RAN proteins which bound to GTP, RAN GTP complex we have here. It is needed to detach importins from this cargo protein that have entered the nucleoplasm. And then so RAN GTP complex together with this importin will detach from that complex, and then alpha and beta importins also will separate it, and that cargo protein enters the nucleoplasm. Afterward, this RAN GTP complex together with importin, it will again return, it will again enter the beta importin will again enter the cytoplasm. In cytoplasm, this complex will dissociate with each other from each other, and then the importin again is ready to carry other substance into the cell, into the nucleoplasm, and this RAN GTP complex will dissociate into phosphate RAN GDP complex, so they will dissociate from each other, and now the beta importin is ready to carry the next protein or other thing into the nucleoplasm. So the transport of substances through the nucleopore complex, there are three types of transportations. First of all, it is passive transport. If the substances has diameter less than 10 nanometers, they may pass without any energy. Then there is facilitated transport, and so in this case the importins have important role. Then we have active transport, just I told you that is the active transportation of the substances because of the energy of GTP. Here will be used GTP molecule. As we saw, it is needed to for recycling of importins that will carry the other components, other substances into the nucleoplasm. The same thing we will see in export of substances from the nucleoplasm to the cytoplasm. In that case, will participate exportins. They also carrier proteins, but their name is exportins, and they will also recognized by the nucleopore complex, and they also will carry the needed substances to the outside, to the cytoplasm. And here also will be used energy of GTP. So that is the active transport. So that's why the nucleopores permit selective bidirectional exchange of small molecules, ribosomal subunits, and other substances between the nucleus and the cytoplasm. And the number and distribution of nucleopores vary according to activity and the type of the cell. So if the cell is active, there are more nucleopores than in the cells which are non-active. And so metabolically active cells will have a lot of nucleopores. The outer rim of each pore forms by fusion of outer and inner membranes, just we mentioned. And in transmission electron microscope, the protein coated gold particles, here we see they passing through the nucleopore complex. So as nuclear envelope impermeable to ions and molecules of all sizes, the exchange of substances between the nucleus and cytoplasm is made only through the nucleopores. So we have to remember it. Then ions and molecules with a diameter up to 9 or 10 nanometer pass freely through the nucleopore without consuming energy. And molecules larger than 10 nanometer or 9 nanometer are transported by active process mediated by receptors, which uses energy of ATP, also GTP. And now we are coming to the chromatin. The chromatin's material is surrounded by nuclear envelope. Actually, chromatin, it is the storage of genetical information, because when we talk about the chromatin, we mean the complex of DNA molecules and the proteins. So chromatin, it is the it means the DNA molecules and also the proteins which are needed for the packaging of DNA molecules. And now we can see the for example, it's specialized stained nucleus under the electron microscope in interface nucleus of a skeletal muscle. Here we can see the nucleolus and the chromatin which is present, which is scattered here. Here the chromatin is network of slender filamentous threads. And in this region, we are looking at the chromatin of dividing cells. So in the dividing cell, the chromatin undergo condensation, supercondensation, and form these threads that are the chromosomes. So supercondensed chromatin forms chromosomes that we can see under the microscope. But in this case, chromatin is scattered. That's why we cannot distinguish individual chromosomes, and that is the chromatin of interface nucleus. But in dividing cells, we are looking at the condensed, super condensed chromatin, which is actually the chromosomes. And now we are looking here also at the chromatin, and the chromatin that are chromosomes, which are free inside of the cytoplasm, because during cell division the nuclear envelope disappears and the chromosomes are free inside of the cell cytoplasm. Nucleus, here we are, the chromatin, it contains DNA and associated nuclear proteins that are necessary for DNA to function. And chromatin has affinity for basic dyes such as hematoxylin used in light microscopy. Because PO4 phosphate groups of DNA of chromatin are responsible for basophilia. That's why it is stained by basic stains. The main characteristic feature of the staining feature is the Fulgen procedure. It is the specific histochemical reaction for desoxyribose of DNA. So Fulgen stain, it is specialized for the nucleus. Fluorescent vital dyes such as Hurst dyes and propidium iodide and DAPI are used for visualization of the nucleus nowadays. These are histochemical stains which we use to visualize the nucleus in immunohistochemistry. Chromatin pattern of nucleus, it is have a different appearance. It depends on the activity of the cells. So the cells with light nuclei as in neurons, are more active than those with condensed dark nucleated ones. If the nucleus is dark, as we are looking here, it means that there is chromatin is condensed and it's not active cell. But in this case, the neuron, it's very active. It has very important, a lot of functions. That's why the nucleus is light in appearance, because it rich in decondensed chromatin. And so more DNA surface is available, because here more DNA surface is available for transcription of genetical information. As we know, the chromatin we have in two forms, heterochromatin and euchromatin. Heterochromatin, it's condensed chromatin. Here we can see dark regions because of the condensed chromatin, and this nucleus include completely condensed chromatin, and this nucleus also. And it means that here chromatin condensed, it's heterochromatin, it's not active because it's condensed. And so this nucleus is characteristic feature of non-active cells. But euchromatin, it's decondensed. That's why it has light appearance and it's present in active cells. Active, it means that cells produce high amount of proteins. For production of proteins, first step, it's transcription, it's formation of the RNA molecules on the DNA template, the transcription process. And so in dark stained nuclei, rich in heterochromatin, coiling of DNA makes less surface available for transcription. Here we can see the heterochromatin, which is present mainly, its main location, it's under the nuclear envelope, heterochromatin. And euchromatin is light and it's mainly located in the center of the nucleus. And here we can see the light appearance of the nucleus because it's rich in euchromatin. And in this case also, so heterochromatin, it's electron dense, coarse granules and in electron microscope, and basophilic clumps in light microscope. Euchromatin is electron lucent. It is a pale areas in nucleus. The proportion of heterochromatin to euchromatin accounts for light to dark appearance of nuclei in tissue sections. Here that is active nucleus, it is inactive because it rich in heterochromatin. So euchromatin is prominent in neurons and hepatocytes, which are metabolically active, and heterochromatin, it predominates in circulating lymphocytes, these are blood cells, and in sperm, densely stained clumps embedded in lightly stained clear areas. And so heterochromatin's main locations, these are perinuclear chromatin, it's under the nuclear envelope at the periphery of the nucleus. Then karyosomes, which are found throughout the nucleus. They are discrete bodies of chromatin, irregular in size and shape. And nucleolar organizing, nucleolar associated chromatin found in association with the nucleolus. And here we can see organization of the chromatin, and it consists of DNA and structural proteins. So how it is organized? Chromatin, it's complex. That is, here we can see that in eukaryotic cells, length of DNA molecule is 100,000 times longer than nuclear diameter. For example, diameter of the DNA is 2 nanometers, but diameter of the nucleus is approximately 6 to 7 nanometers. So 6 to 7 micrometers. And how? But length of the DNA, all of the, if we will join all of the chromosomes to each other, we will see that the lengths there are approximately 1.5 to 2 meters. How that 2 meters long structure may be located within the small nucleus, which have 6 to 7 micrometers diameter. So that's why we need some packaging mechanisms that will package DNA molecules within the nucleus, and that's why the DNA is highly folded and tightly packed in formation of unique nucleoprotein complex, that is we call the chromatin. That's why it have to packaged and to form chromatin. Further folding of chromatin occurs during mitosis that produce chromosomes. So chromosomes we can see just at the cell division. Here we are looking that that is DNA molecule, DNA double helix with diameter of 2 nanometers. Then the DNA molecules will form some structures which we'll call the nucleosomes. The nucleosomes, these are rounded structures which 11 nanometer diameter, 10 to 11 nanometer in diameter. And nucleosome, it means that there is the structure which have core that composed of the proteins, histone proteins. There are 8 histone proteins, 4 in this side and 4 in opposite side. They form core, and around that core the DNA forms these turnings, 1.65 times in nucleosomes. Then these free ends of DNA will join each other at the H1 protein, that is another histone protein that is yellow colored here, and it will form two. So by that way there is formed chromatosomes. So from the nucleosomes is formed the chromatosomes. Here around the core will be the DNA forms two turns forms here, and here we also have linker region of the DNA. So by that way the chromatosomes are formed, and they will resemble beads on a string. Beads on a string, it means that the beads are chromatosomes, but string, this is the DNA molecule which join these successive chromatosomes to each other. Then on next step, the chromatosomes, 6 to 8 chromatosomes, they join each other and form these rounded structures like flowers, or like flowers, yes, like leaves of the flowers. And these structures will have diameter 30 nanometers. These structures called solenoids. Then the solenoids here, they are more in diameter. They undergo coiling, turning, and they form these chromatin fibrils. These fibrils form loops, and the loops then will have more diameter. They are hierarchically turn, and by looping they increase in diameter. They become 30 nanometer in diameter. Afterward, these loops will join each other by another protein that will be called chromosomal core protein. And in the center of this structure, we will have another protein. It is non-histone protein that is called chromosomal core protein. And finally, by this condensation, supercondensation, we will have chromatids. One chromatid is one DNA molecule. Another chromatid here, another DNA molecule. They will join each other at the, so they will form sister chromatids, and they join at each other at the centromere region. Diameter of each chromatid is approximately, it will be 700 nanometer, and the diameter of all chromosomes will be approximately 1400 nanometer. Here we can see the all structure, all of them just I mentioned, that is the nucleosome, the central core of the nucleosome with histone proteins, histone H2A, H2B, H3, and H4. They form this octamer core around which DNA turns. When it forms two turns, here they join at the H1 histone protein. So that is a nucleosome or the chromatosome. Further, it says it has 10 nanometer diameter. And additional 48 base pair segment forms a link between the adjacent nucleosomes. So that will be link region with 48 base pairs of the DNA molecule that will they will join, and they will connect neighbor nucleosomes. And so here we can see also these nucleosome regions. First of all, in the first step, the 1.65 times around the histone octamer core, it is the nucleosome. And then when there appears H1 linker protein, there is two turns, and so it's wrapped around this core, DNA wrapped around the core. And here we can see chromatin fiber formed by the linear alignments of the nucleosomes. Here nucleosomes, they are like beads on a string. Finally, we can see this our chromosome. Chromosomes we can see just at the mitosis and at the prophase and at the mainly at the metaphase of mitosis. Mitosis or meiosis, it's cell division. Here we can see the same thing, DNA, then nucleosome, chromatosome, beads on a string, solenoids, loops, and central core proteins. Here together they will form chromosomes, chromatids, one chromatid and neighbor sister chromatids, centromere region. And here we are looking at the kinetochores. At the free ends of the chromatids, we have telomeres. Telomeres, they are needed for structural integrity of the chromosomes. And so, along with other non-histone proteins, DNA molecules, and all of the structures just I mentioned, you can see here. So during mitosis and meiosis, there are higher orders of coiling, condensation of chromatin fibers formed from chromatin loop domains attached to a flexible protein scaffold, and they form chromosomes. Here, you know that the main function of the DNA is function what we have in our cells, that is a transcription. It's formation of the messenger RNA from the DNA scaffold, and that is a direction of RNA synthesis. Here, by the RNA polymerase, the enzymes participate in this process, and that is chromatin fiber, and that is direction of RNA synthesis. And from the DNA, first of all, there is formed pre-RNA molecule, and it's how the ribosomal RNAs are formed. Ribosomal RNAs, we know that we have 5S, 5.8S, 18S, and 28S. From the previous lecture, we know that ribosomal subunits, they are made up of ribosomal RNAs and the proteins. And ribosomal subunits are formed in the nucleolus. Because in the nucleolus we have the regions of DNA molecules which carry information about these ribosomal RNAs, and when the transcription is taking place, from the DNA is formed pre-rRNA molecule that include 18S, 5.8S, and 28S ribosomal RNAs. But 5S, information about the 5S rRNA is written on not on the DNAs which form nucleolus, but on different chromosomes. That's why 5S will be produced in it derives from another gene, and that's why it will come from other region of the nucleus, and it will also incorporate it into the large rRNA molecule. Then this large molecule pre-rRNA will be processed and will be cleaved into different regions, and then they are surrounded by the proteins, and the ribosomal subunits are formed. Then they are transported through the nuclear pore complex into the cytoplasm to be able to participate in the protein synthesis. The chromosomes we have of different types according to location of the centromere region. We have metacentric, if as we are looking here, the P arm and Q arms, if they have the same size, that is a metacentric chromosome. If there is one of them P arms shorter, submetacentric. If there is significant difference in these arms, that is the acrocentric and telocentric chromosomes we have. And so the telomeres, just I mentioned, telomeres, these are areas that located at each end of the chromosome. Its length is important indicator for the lifespan of cell, and by the length of the telomere we can talk about the age of that cell, about the age of the person, because telomeres they become shorter with each cell division. So with increasing of age, the length of the telomere decreases. And human somatic cells, actually we know that have 46 chromosomes organized into 26 homologous pairs. Each chromosome in the pair has the same shape and size. 22 pairs have identical chromosomes. Each chromosome pair contains same portion of genome and are autosomes. And 23rd pair of chromosomes are sexual chromosomes. and this designed as the X and Y chromosomes. Here the chromosomal appearance under the microscope. So these are the chromosomes, and in the dividing cell we have subdivision of the chromosomes into individual chromatids. So chromatids will detach from each other at the cell division because centromeres will degrade, undergo degradation, and chromatids will separate from each other and they will move to opposite poles. Here we can see the anaphase stage of the cell division, mitosis. Here we also can see it. And that is metaphase, when the chromosomes align in the equatorial plate. That is spindle apparatus, mitotic spindle apparatus, and these are the centrosomes, which are located at the poles. And karyotype, that is a number and characteristics of the chromosomes encountered in an individual. In a karyotype, chromosome pairs are sorted according to their size, shape, and emitted fluorescent color or stained with giemsa. Giemsa stain is widely used to color the chromosomes. And nowadays is very widely used FISH methods, that is fluorescent in situ hybridization methods. And that's chromosomes that stained by these methods, FISH methods procedure. They captured images and are processed by the help of software according to their morphology to form a karyotype. Here we can see by FISH method staining by the FISH method, by in situ hybridization methods. And here cells, there are some steps in this staining methods. First of all, cells, they induced to divide by using phytoagglutinin in cell culture, and then they arrested at the metaphase of mitosis by colchicine. Then cells are immersed in hypotonic solution, causing swelling, rupture, and investigating pattern of chromosomes onto the slides. Chromosomes are grouped according to cell size and morphological characteristics into 23 numbered pairs. Then karyotypes are used for prenatal determination of sex in fetuses and for prenatal screening for certain genetical diseases. And the sex chromosome, what means? Actually, we know that there are, in female cells, there are two X chromosomes, two sexual X chromosomes. And one of these X chromosomes in female, it is always is present in inactive state. That one we can see in some cells, which in those cells it's more pronounced. And that region, that chromatin of the inactive X chromosome, is called bar body. And that bar body we can distinguish in the neutrophils of the blood and in the epithelial cells of the oral mucosa. That in those cells, bar body is more pronounced, and we can see it as drumstick. It has appearance of drumstick-like structure, and so that is a sexual chromatin. Here we can see the drumstick in neutrophils and in buccal epithelium. So in the oral cavity epithelium, we can also distinguish that bar body in epithelial smear, which is prepared from the oral mucosa. And here the nuclei in different stages of the cell cycle. And now we are going to the cell cycle. Nucleolus, that is the region of the nucleus. It is spherical, non-membranous structure. It is site of rRNA synthesis and initial ribosomal assembly, just I mentioned about it. And it's rich in rRNA and proteins. And it has basophilic appearance with hematoxylin eosin staining. It varies in size but well developed in cells active in protein synthesis. Why? Because if the nucleolus it produces ribosomal subunits, so the cells which active in protein synthesis, they require more amount of ribosomes. That's why that nucleolus will be more active in those cells, and those cells may have even not one but two nucleoli inside of the one nucleus. And so rRNA are synthesized and modified inside of the nucleus. The nucleolus has some regions. The central, it is nucleolar associated chromatin. It is fibrillar center. And this from one to several pale staining regions are composed of these are pale regions, which composed of the DNA loops of 5 chromosomes, 13th, 14th, 15th, 21st, and 22nd chromosomes participate in the formation of the nucleolus. Why? Because they have these regions, nucleolar associated chromatin regions of DNA that encode ribosomal RNAs. So it is the fibrillar center of these sequences of bases code for rRNA. And so that is this. Then there is pars fibrosa. Pars fibrosa, it contains ribosomal genes that are actively undergoing transcription, and large amounts of rRNA pre-transcripts are formed from the DNAs. They form pars fibrosa. Then pars granulosa consists of the granules. These are maturing ribosomal subunits. And here the ribosomal subunits assembly, and they form particles. Then they will go out from the nucleus and enter the cytoplasm. Nucleonema, the network formed by granular and fibrillar materials. rRNA is present both in granular and fibrillar material. So in granular and in fibrillar, in both of them, there is a rRNA molecules present. Here we can see the nucleolus and how in the nucleolus produced these ribosomal subunits, and how they will go out from the nucleus. Dense fibrillar component here, granular component here, and fibrillar center here. Nucleolus under the light microscope. Appearance here. Nucleoli appear Fölgen negative with Fölgen positive nucleolus associated chromatin. They are methylene green positive. That often rims the nucleolus under the light microscope. Nucleolus is involved in regulation of the cell cycle. Actually, nucleostamin is a protein that has been found within the nucleolus. Nucleostamin is a p53 binding protein that regulates the cell cycle and influences cell differentiation. So it is present in the nucleolus. Pay attention, nucleostamin and p53 proteins. They are present in nucleolus. These are components which control cell cycle and prevent formation of the tumors. If the damage of the DNA we will have, so the cell cycle have to be stopped. And that mechanism for stopping cell cycle with damaged DNA, it is the responsibility of this p53 protein, and together with the nucleostamin. So the nucleolus also participates in the regulation of the cell cycle and differentiation. Nucleolus we can see here also. Nuclear matrix, it fills the space between the chromatin and nucleoli in the nucleus. So that is the It's composed of mainly proteins, metabolites, and ions. And nuclear lamina, which is located under the nuclear envelope, it is also part of the nuclear matrix. Besides, the nucleoplasm has nuclear skeleton. If the nucleic acids and other soluble components removed, so the fibrillar structure remains, and that is called nucleoskeleton. And it contributes to the formation of protein base to DNA loops, and so that is the base for the binding. Here we can see blood smear. It's unnucleated erythrocytes, so that's cells which do not have any nucleus. Then we can see nucleated leukocytes, and they are polymorphonucleated cells, neutrophils, which have polymorphonucleated. They are polymorphonucleated because the nuclei is lobulated. There are 3 to 3 lobes. And also we have mononucleated cells. These are lymphocytes. Liver cells, the nuclei we are looking here. Then the cerebellum, Purkinje cells, which have, we can see their nuclei. Multinucleated cells, for example, they have a lot of nuclei. And the cell cycle. The cell cycle, it represents a self-regulated sequence of events that controls cell growth and cell division. So it's a series of subsequential cellular events in preparation for cell division. And it is composed of the interphase, when the cell becomes larger and duplicates its genetical material, and mitosis, a process that results in the formation of 2 daughter cells, identical cells. The cell cycle is usually described as beginning at the end of the cell division, when the cell is entering interphase. And here the stage of cell cycle. The interphase is also subdivided into G1, S, and G2 stages. Certain cells that are highly differentiated, for example, muscle cells and neurons, they cease to continue and to go through the mitosis and remain in a resting stage, G0 stage. Other cells, such as peripheral lymphocytes, enter G0 phase temporarily, and at a later time they may again enter the cell cycle. Events such as mechanical forces, ischemia, and death of cells in a particular cell line may induce signaling cells to release growth factors that induce the expression of proto-oncogenes, and which prompt the proliferative pathways of the cell. This process activates the release of cascade of cytoplasmic protein kinases that triggering a series of nuclear transcription factors regulating the expression of proto-oncogenes that result in cell division. And many cancers are the result of mutations in the proto-oncogenes that permit the uncontrolled proliferation of the mutated cell. Here we have to mention the group of proteins known as cyclins. By complexing with specific cyclin-dependent kinases, that they not only activate them but also guide them to target proteins, and in that fashion control the entry and advance of the cell through the cell cycle. So there are checkpoints that control and prevent the cell from entering or continuing the cell cycle if there is something went wrong. So at the checkpoints the cell is checked, and so we have to know that pay attention that cell cycle stage, the mitosis here, G1, S, and G2. So here we can see the checkpoints and there are several checkpoints, approximately 5 main checkpoints we have, and in these checkpoints we have also 3 checkpoints for controlling the DNA molecules if there's in the DNA there is a damage. If the DNA is damaged, the cell cannot continue through the cell cycle and it has to be stopped. The cell with damaged DNA, it has to be stopped. And so the passage of the cells through these checkpoints, it is the that is the function of the cyclin and cyclin dependent kinases. For example, let's look here. In G1 stage we have a restriction checkpoint, and this restriction point, it is in gap 1 in G1. It permits the chromosome duplication and the entry into the next stage. And here the cell is checked for some requirements. We know that in G1 stage, it's at the conclusion of mitosis, the newly formed daughter cells enter the G1 phase of the cell cycle, and it is here in this stage they produce regulatory proteins necessary for DNA replication, and they restore the nucleoli and the original cell volume that was before cell division. We know that cell division produced 2 daughter cells with decreased volume. That's why the cell will restore its volume, its DNA, its proteins, and all of this, and plus, and in addition to these processes, the acceptance of the mitogenic signal is controlled at this point, at this restriction point. That's why if the cell have not produced enough proteins and so on, the cell cannot continue this cell cycle and it will be stopped here. It may die by apoptosis, programmed cell death, if those problems will not be fixed. Or it may enter the G0, it may leave cell cycle. Then we are coming to the end of the G1 and there is a DNA damage checkpoint. Then we have DNA checkpoint at the S stage and in G2 stage, and that is DNA checkpoint. And in addition, in G2 stage we have also checkpoint for unreplicated DNA. If in the S stage, we know that S stage is characterized by the duplication of the DNA molecules. If there is problem in duplication of even one chromosome, the cell will not pass through this checkpoint and it will be stopped till that problem is fixed. Then it may continue cell cycle and enter mitosis. In mitotic division, we have also at the metaphase, we have spindle assembled checkpoint. If all of the chromosomes will not come to the equatorial plate, which is requirement for the metaphase, the cell will stop. So that are the checkpoints, and those checkpoints are controlled by CDK cyclin complexes. So CDK there are 4 classes of cyclins and CDKs with which they form complexes, and in this one we have to mention that passage of the cell through, for example, G1 cyclins, in G1 operating cyclins, this is a cyclin D together with the CDK 4 and 6, or they will bind to each other and they will control passage of the cell through the restriction point. And then in G1 S cyclins we have that is the cyclin E together with the CDK2, they bind, and these 3 complexes, and they along with other intermediaries, they permit the cell to enter and progress through the S phase. And then we have cyclin A that binds to CDK2, forming complex that permit the cell to leave the S phase and to enter the G2 phase and induce the formation of the cyclin B. It induces the formation of the cyclin B. In mitosis, we have cyclin B that binds to the CDK1. It is cyclin B actually here. It binds the CDK1, and this complex allows the cell to leave leave G2 and enter the mitosis, to the cell division. And S stage is characterized by the application of DNA and centrioles also. G2 stage is characterized by production of the ATP, that reserves are increased, and the newly synthesized DNA is checked for the possible errors, and if present is corrected. So that is processes, and that are the CDKs that I mentioned, each of which is active in which stage. So CDK1 and cyclin B, it is a main complex which controls the cell through the passage of the mitosis, the cell division. And here in G1 stage, as I mentioned, that there is restriction checkpoint, and this checkpoint is mediated by the interactions between retinoblastoma susceptibility protein and a family of essential transcription factors with target promoters. When the retinoblastoma and transcription factor 2F form a complex, the transcription stops. So that is the complex which will prevent passage of the cell through the restriction point. So retinoblastoma susceptibility protein and essential transcription factors E to F. If there is some problems in the genome, in the genes that encode to this component, these substances, so the cell will not be stopped at the restriction point, and it will continue through the cell cycle, and it may create tumors, cancers. And we have to know that many cancers are the result of mutations in the proto-oncogenes. So just I mentioned these proteins and transcription factors. These are result of the transcription of those genes which are called proto-oncogenes. And if there is mutations in proto-oncogenes, it permits the uncontrolled proliferation of the mutated cell that finally lead to cancer. So mitogen signals that cells have to receive to enter the cell cycle. These are growth factors, hormones, cytokines, neurotransmitters, adhesion proteins, macromolecules of intercellular matrix, physical factors. All of them, these are mitogen signals. The mitosis. We have two types of cell division. It is the mitosis and meiosis. Mitosis is division of the somatic cells. It has some stages. It is a prophase where taking place condensation of the chromatin, and we can see individual chromosomes inside of the nucleus. And then at the end of the prophase, nuclear envelope disappears, nucleolus disappears, and there is forming of the mitotic spindle apparatus. The centrosomes move to the opposite poles of the cell, and between the centrosomes formed mitotic spindle apparatus. Then chromosomes at the metaphase, they come to the equatorial plate, and then here, the centromeres disappear at the anaphase, because anaphase promoting factor we have that will activate securin, and it will activate separase enzyme that will break centromeres, and the chromosomes will subdivide into sister chromatids. Each of the chromatids will move to opposite poles of the cell at the anaphase, and at the telophase the cells will restore the self, they will restore nuclear envelope, and mid body is formed in the center. Pay attention. So these are stages: metaphase, then anaphase, and telophase. in the center of the cell is formed mid-body. There is actin myosin complexes that bind to the cell membranes, and between them taking place constriction, and in the center we have these fibers of the mitotic spindle apparatus. These are microtubules actually. Then they degrade, disintegrate, and finally by activity of the actin myosin complexes, the membranes opposite sides move toward each other, then they fuse and the cell subdivides into two. Here our cells in the G1, S, G2 stage, that is the interphase, that is the mitosis, and here we can see the meiosis, meiotic cell division. Mitosis just I mentioned, and as a result of mitosis there is formed daughter cells, each of which include 46 chromosomes. The number of chromosomes is identical to the mother cell, but amount of chromatids is less. So in this case, then each chromosome includes just one chromatid in these cells. That are one chromatids. These are inside of the chromosomes. But in meiosis it is division of the sexual cells, and it includes two subsequential divisions. That is the first meiosis or equation. It is reductional division, and equatorial division. In first case, in the first stage of the meiosis, we have the prophase is subdivided. Pay attention. That is prophase 1 of meiosis. It's subdivided into some sub-stages: leptoten, zygoten, pachyten, diploten, and diakinesis. Here taking place condensation of the chromatin. Chromosomes are formed. Then these homologous pairs, they come to each other, and these homologous chromosomes align in gene-to-gene register to form synaptonymal complexes. And in pachyten, homologous chromosomes continue to condense, and crossing over sites, chiasmata, are formed between the non-sister chromatids, and it results in random exchange of genetical material here. It is a genetical recombination is taking place here, and then we are coming to the diploten. In diploten, chromosomes continue to condense, followed by disjunction of the homologous pairs. So they begin to detach from each other, and as the chromosomes condense maximally in diakinesis, the synaptonymal complex disassembles, the nucleolus and the nuclear envelope disappear, and chromosomes are now free in the cytoplasm, and a microtubule spindle begin to form. Now we are coming to the metaphase 1. Homologous chromosomes align in random order on the equatorial plate, ensuring a shuffling of the material and paternal chromosomes. The kinetochore microtubules attach to the kinetochores. So here we have kinetochore microtubules that attach to the kinetochores of the chromosomes. In anaphase 1, homologous chromosomes still composed of the two sister chromatids, they migrate to the opposite poles. Here we are looking how they move to the opposite poles. And telophase 1 of meiosis 1 is similar to the telophase of mitosis, but chromosomes complete the migration to opposite poles, and nuclei are reformed, and cytokinesis divides the cell into two daughter cells, each of which has the 1n number of chromosomes, 23 chromosomes. That's why the cells which are formed as a result of first division of meiosis, they have just 23 chromosomes. That's why this division is called reductional division. It is reduction of the number of the chromosomes. Then meiosis 2, it is equational division. Equational division is not preceded by another S phase. So there are not S phase, just after the first meiosis comes the second meiosis. It resembles mitosis, and it also has prophase, two metaphase, anaphase, and telophase. Chromosomes still composed of sister chromatids become arranged. Let's look here. And so the meiosis actually will produce haploid set of chromosomes. That's the gametes, our sexual cells, divide by that division type. And here we are looking at the meiosis 1, how the synaptonymal complexes are formed, recombination takes place, anaphase, the division of the cell into two, that's first meiosis, and that is second meiosis, by which the cell subdivides again into two, but here it's like mitosis, in which chromosomes will subdivide into individual chromatids. That's why into the pools of the cell will move chromatids. That's why, as a result of meiosis 2, formed cells, dotted cells, each of which include haploid set of chromosomes, 23 chromosomes, and each chromosome has just one chromatid. Here we can see appearance of synaptonymal complex that is one chromosome, another chromosome, and that is a complex which is formed between them to subdivide for the recombination. Here will take place the genetical exchange of materials of genes between the homologous pairs. Polyploidy, it is when the mitosis and without cytokinesis, it results in the formation of cells with twice as many chromosomes. So if the process of division of the chromosomes, it will be interrupted. In one cell may be present one more chromosome, and in another cell may be present one less chromosome, and that will lead to the problems. Or we have polyploidy, in which one item that may be absorbed from karyotype is aneuploidy, it's missing one chromosome, and or there may be one more chromosome. It is trisomy. For example, Down syndrome, people with Down syndrome have extra chromosome 21, and that's called trisomy 21, and they exhibit mental retardation, stubby hands, many congenital malformations. Klinefelter syndrome, it's when there are 3 sexual chromosomes, but Turner syndrome, there is missing one sexual chromosome, and there will be X0. All of them lead to problems in mental retardation and in physical problems, and those persons cannot live long. And cell death. Cell death, it is the cell death. It has 2 main types. When cells die because they no longer receive nutrients or are exposed to sudden trauma, they undergo necrosis, a process that initiates the inflammatory response. So in this case, cell membrane integrity is interrupted, and a large amount of fluid enters the cell. It leads to burst of the cell, osmotic burst, because high amount of fluid will inside. Then lysosomal enzymes are activated, and finally it is the breakdown of the cell, and it will initiate inflammatory response around. But in another type of the cell death is a programmed cell death. In this case, it is the apoptosis called, and in this case, it's programmed, genetically programmed cell death. Here it may take place because of the specific environmental conditions such as overcrowding, or it is because of the age, increased age may lead to apoptosis, or virally transformed cells. They also forced into apoptosis by cells of immune system, and or in actually in embryonic development there is taking place dying of many neurons, nerve cells, or other components which are not needed for the human body. So that all of them, these are examples of cell death because of that programming at the genetical level. And apoptosis is characterized by the condensation of the nucleus, condensation of the chromatin, then fragmentation of DNA molecules, then fragmentation of the cell cytoplasm, and finally the cell subdivides into individual fragments, each of which surrounded by the membranes. That's why we cannot, the apoptosis do not lead to the inflammation, because apoptotic bodies we can see here, each of them, they have the membranes that surround them and isolate them from surrounding environment. But on the surface of these apoptotic bodies arise signals that will be recognized by the neighbor cells of the immune system and or macrophages, and they will be swallowed up. So that is all for today's lecture, dear students. This was the series of lectures on the cytology, and see you on next lectures. It will be the lectures on the embryology.